just says the system's warming up. Thanks.
So did that work, sir?
Yeah, thanks a lot. It does work now. Thank you. Okay, folks, if you don't mind, I'm going to try to get started. So we're going to go back and start talking a little bit more about the mechanisms that we can use in this gene drive that I mentioned to you before. So it is actually not an academic exercise. This is a real question that is being asked not only in parts of Brazil and Colombia, but also in the United States. As Zika is now moving into both Texas and into Florida, in different regions of the United States, different regions, districts, etc., are seriously considering the release of genetically modified organisms to combat Zika from spreading any further. And this story is actually a really interesting one. And this CRISPR-Cas9 system that I want to outline for you today, and I want you to take away from today's lecture all of the technical aspects of what CRISPR-Cas9 is and compare it by the end of today's lecture to some of the other genome editing tools that we've had in the past. And you might wonder, if genome editing tools have been around since the 1980s, why is everyone still fascinated by this CRISPR-Cas9 system? I want you to be able to tell me by the end of today's lecture why we are so interested in this technology, how it works, what are some of the technical considerations for this. As some of you will know, right now, when we talk about CRISPR-Cas9, it's really hard to get away from the individuals that have started the whole story with CRISPR-Cas9. And the individual here on the right, this is Dr. Fang Zhang over at MIT, and two other individuals, Dr. Charpentier as well as Jennifer Doudna in the middle, represent the three individuals that everyone recognizes as being involved in CRISPR-Cas9 technology. Now, the big issue is, who actually developed CRISPR-Cas9? Is it really Fang Zhang? Because at MIT, they believe that he did. He's the person that came up with the technology that we use now. is it really Emmanuel Charpentier as well as Jennifer Doudna, as people in California believe? And right now there's a battle between the two different universities, MIT versus the universities in California, because there's a lot riding on this. Whoever wins this war related to the patent, and whoever owns the technology is really going to own genome editing. And if for you to be able to dictate who gets to use or who gets to say that you own genome editing is really one of the biggest issues right now in terms of biology. So I'm going to take you through some of the different aspects of CRISPR-Cas9, why we're interested in it, go back and revisit different aspects of gene drives, examine some of the issues that, technical issues that are involve some of the gene drive elements. I'm going to introduce you because Fang Zhang actually comes from a neuroscience background. And he's actually the co-inventor of not just sort of the CRISPR-Cas9, but other core neurobiological techniques like optogenetics. Some of the issues that are associated specifically with neurobiology and genome editing. And then we'll go over some of the advantages and caveats. So one of the reasons why we're so interested in gene drives is that when we're talking about sexual reproduction, as I mentioned in the last lecture, one of the things that normally happens is that there's a 50-50% chance that a particular trait, gene, etc., is going to be inherited by the offspring from their parents. The gene drives allow us to manipulate the genome, but if we do nothing more than manipulate the genome, make a correction, make an alteration, then it's still going to be passed along according to normal Mendelian inheritance. If we also use a gene drive with it, we can actually enhance the likelihood or the probability that that gene that we've altered or edited is also going to be passed along when that organism undergoes sexual reproduction. And we know that this happens in nature. So one of the most commonly used genetic organisms is Drosophila. And if you look at Drosophila, one of the things that was found out in the 1950s. We started using Drosophila as a model organism in about 1905. It was one of the first reports, and now is a very commonly used animal organism. One of the things that we know in the wild, compared to different species that were kept in the lab, in the wild, the Drosophila actually started to have these gene drives to developing P elements. And this was happening in the wild. It was not happening in the lab. And this was one of those really interesting things that was found in the 1950s, that somehow in wild type species, not the ones that we were rearing in the lab for experiments, that this transposable P element that allowed mutations to be inserted in the genome was occurring out there in the wild, but not in the lab. And so we became really interested in this concept of this gene drive. And we knew that it can actually happen in nature, especially using transposable elements to cause mutations in certain genes, as we're finding in Drosophila. Every sexually reproducing species has some element of this type of gene drive associated with its genome. So we know that it does happen. We know that in theory, we might be able to use gene drive to eliminate different diseases like dengue or Zika, etc., or the vectors that carry them. We're not sure how best to use this right now. It's still an ethical debate. Do you wipe out an entire species from one particular region? And if it's a rapidly reproducing species like mosquitoes that has an average lifespan of somewhere around 7 to 10 days, you can wipe out that species over the course of a few years. And that species, in theory, would not really come back. And is that something that we want to be able to do? We could, for example, right now in Toronto, one of the things that we're really worried about is that there is an invasive species that's been coming up from the United States toward Lake Ontario. And we know that an invasive species like the Asian carp, for example, if it made its way into Lake Ontario, would be very deleterious. So should we, in theory, be thinking about wiping out this non-native species that can outcompete the normally occurring fish in our own lakes before it actually reaches us here? We know that if you're living up in the Markham, York region area, that a lot of your elm trees are gone now because the longhorn beetle has decimated that population of trees? Should we eliminate them to save our economy, to save our woodlands, etc.? Again, a lot of ethical debates as to whether or not gene drives should be used and introduced into certain species to completely eliminate them. We know that there are also some other issues that we're looking at. So we've caused problems in the past. We've used pesticides, and now we know that many of the different insecticides, pesticides, all of the organisms that we originally targeted are actually now resistant to all those pesticides. We know about antibiotic resistance. Should we go in now and use genome editing tools to actually reintroduce genes that make these different populations more susceptible to the different types of toxins that we release? So for example, glyphosate is commonly a compound that is used to control weeds. Those weeds that were originally knocked out by glyphosate in the 1970s and 1980s, those weeds are no longer susceptible. You can't actually destroy weeds anymore with glyphosate, and as a weed killer, it doesn't work very well. And so can we go back and re-engineer something that will make these weeds that we see now more susceptible to things like glyphosate? So one of the reasons why we're so interested in this CRISPR-Cas9 technology is really the issue with this genome editing. If we can do all these different things, wipe out non-native species to make sure that these issues with different types of resistance get overcome, we need a way to do this type of genome editing rapidly, quickly, as well as cost efficiently. And one of the things that is really remarkable about this CRISPR technology, and I gave you a quote at the end of Monday's lecture, is that even a third grade student, in theory, can do this type of technique. It's not a complicated technique as long as you have some ways in which to manipulate the genome. We know, for example, that this technique has been actually in use for a long time. And unfortunately, I'm a little under the weather today. I would have brought in some food samples for you. Every time you eat a piece of cheese, or every time you eat yogurt, you are actually making use of CRISPR-Cas9 technology. One of the reasons why we know that CRISPR-Cas9 exists, and we've been using this ourselves for a long time. If you've ever gone and visited a cheese factory or a yogurt factory, all of those rely on bacteria to ferment your cheese as well as your yogurt. The bacteria themselves can become infected with viruses, the same types of viruses that we've been talking about with Zika. In order for those bacteria to fend off the different viruses that normally infect them, they're called bacteriophages. One of the things that they do is to take the bacteriophage DNA, the viral DNA, and to chop it up using CRISPR. It stores it sort of as a memory, almost like an immune memory of that, of the virus that infects it. And why do we care about that? Because when you infect a bacteria with a phage, the way in which it causes fermentation goes off. The cheese doesn't taste as good. The yogurt tastes more sour. And so what individuals did was to actually inoculate cultures with bacteriophage, get the ones that were resistant. They didn't know how it was resistant to that, and that retained its flavor making capacity to bypass this whole system. Later, much later, it was found that these bacteria that were resistant to this bacteriophage infection actually had CRISPR enzymes that were involved in storing the bacteriophage memory that allowed them to become resistant to viral invasion. So this whole idea that CRISPR bacteria can actually cause the elimination of genomes through this nuclease known as Cas9 was really important to us. We didn't understand how we would use it way back in the 1980s, and I'm going to show you that our understanding of the technology has increased dramatically, but there are a lot of other things that I want to go through as well. So a couple of things before I get to that point about why we're so interested in this technology from the 1980s, late 1980s, 1990s, and onwards. So CRISPR is not going to work everywhere. So it's going to work best in exactly the same issues that I mentioned to you earlier in species that reproduce sexually. Okay, so when we are looking at a gene drive and when we're looking at different types of ways in which we can use CRISPR best, It will target insects, it will target animal cells, it will target many plants, but not all plants, as I'll get to in a moment. You also have to take into consideration some of the time that's going to be required for this CRISPR to actually work in a gene drive system. So in humans, for example, even though in theory it's possible, because we reproduce sexually, that it's possible for us to cause a deleterious change in our genome that we're not aware of, but it would take many, many, many generations, hundreds of years before our species got wiped out. That's not the case for short-lived life cycle species like mosquitoes or other types of plants. So we need to take into consideration a lot about the different species that we're targeting, and there has to be efficient mixing. In other words, the more of this altered gene that we can alter using a genome editing tool like CRISPR-Cas9 that we introduce to begin with, the more mosquitoes that we genetically modify and introduce to begin with, the better off this gene drive is going to be. So again, not all plants, for example, most modern crops are not actually grown from seeds anymore directly. They're grown from seeds that are stored in warehouses. So you're not going to genetically modify our crops because the crops are already genetically modified to begin with. So all of these different types of gene drives have to be put into the right type of context. Same thing for most of our domesticated animals as well, our cattle, our food supply. We don't really rely on things like gene drives because we've already manipulated their genomes and we manipulate how they reproduce in very, very structured ways. So one of those big issues that many people have with gene drives is, are we going to be damaging our food supply? I would say and argue, using the same sort of white paper that many of these points come from out of Harvard, that our food supply is probably not at risk. It's more likely that the ecological balance that we're not aware of by disrupting the life cycle of a mosquito and how the mosquitoes might be a food source for other insect species that might play a role in plants is more likely to cause an issue than a direct effect on our food supply directly. So this is one of the reasons why there's little worry or concern about our food supply in general. Okay, so this gene drive also has to be able to cut the target gene. Most genome editing tools that I'm going to show you, whether it's zinc finger nucleases, TALENs, or CRISPR-Cas9, for the most part can target almost every gene that we know of, some more efficiently than others. So we don't really have to worry about that. I'll talk about one other technique, the CRE-LOX system, and even there, the gene drive in theory, if it's using CRE-LOX, could also do the same types of things. We shouldn't be able to alter the ability for the organism to reproduce. It has to be copied well, and these are all the limitations and considerations that we take into account. And again, these are sort of white paper think tank considerations that I'd like you to make sure that you're familiar with for your final exam as well, that some of these considerations for when we do a gene drive that we have to take into account all these different points, that it has to also be stable, etc., is something that we have to think about before we actually do the gene drive. Okay, so again, in terms of targeted genome editing, this involves either manipulating DNA or RNA. In theory, RNA is also a form of genome editing as well. And this could be, in theory, anyways, for rectifying, replacing, or deleting a gene that might be causing a disease. It might be used to wipe out an organism. It might be used to combat infectious diseases like Zika, etc. There are many, many potential uses. As long as you know the gene that you want to target, in theory, gene drives can allow you to do almost anything, at least in theory. The practicality of it is something that I'll talk to you about in a moment, but this is the promise of what gene drives, genome editing is really all about. So again, you don't have to worry about all of the different numbers on this, or the dates that are found on this particular slide, nor do you have to worry about all of the information here. It's really just to give you sort of an overview. When we start off with genome editing, where people started to think, can we actually manipulate the genome? These ideas have been around for a while, and you kind of wonder why CRISPR biology, why it's so important. And again, in the 1980s, CRISPR is identified. We know CRISPR exists in bacteria. We have some ideas what it's doing, but no one did very much with it. Look at the timeline at the very top. People published that in the 1980s, and it's not until the mid-2000s that we actually start having different individuals starting to look at the technology in different types of ways. Now, when we get into the early 2010s, then this whole area starts to explode. And one of the reasons why it actually starts to explode is this one paper here. And this one paper is actually the crux of this entire argument. In 2013, Fang Zhang's group actually came out with one of the most cited papers in CRISPR technology. And he actually showed and demonstrated that in a eukaryotic system, in a mammalian system, that this bacterial system could actually be introduced and alter the genome. And since then, you can see the dramatic rise in the number of papers that get published. Now, because this in theory can be used for a gene drive, since CRISPR, which I'm going to outline for you in the next few slides, can be used in a gene drive, should we actually be worried that gene drives will actually be something that could be used in the germline and to alter embryos? And that's sort of the big bioethical issue right now. In Canada, it is actually illegal to work on CRISPR to actually introduce any changes to the germline at all. In the United States, the regulations are not quite as restrictive as they are here, but you can see that there are many, many other countries that don't have the same sorts of regulations that we have here in Canada. For example, Brazil is rethinking some of these different rules and regulations. And you can see that there are many other countries across different parts of Africa, Asia, as well as in Europe that have very different types of regulations. So not everyone has signed the accord that says that we should stop working on the genome editing at the germline level. So you can't pass on a trait, for example, of everyone having blue eyes after several generations in terms of genome editing. So should we be worried? I would argue again, right now, not really. We don't think that people are actually working on germline editing. There's only been one published paper of germline editing that was in a scientifically peer-reviewed journal, but it might be a concern maybe in about 2 or 3 years' time where it will become revisited. So the next few slides are more technically oriented, and I'd like you to be really comfortable with a couple of different elements. And throughout all of this, I want you to be comfortable with 3 different things. One, what is the difference between some of the genome editing tools? What are the core differences? Some of them use different types of nucleases, so would you be able to identify them? Some of them require very, very specific guides to the DNA sequence that is going to be cut, and do you know what some of those different types of guides are? And then in addition to that, there are also some limitations that are associated with some of these different genome editing tools as well. So the major ones that I'm going to be talking to you about, and 2 of them look very, very similar. They're not too dissimilar, but there are some things that I'm going to point out to you. CRISPR-Cas9 that we'll spend the most amount of time on, which can cut DNA precisely, as well as talins here in the middle, and then zinc finger nucleases here at the very end. And I'm also going to introduce to you Cre-Lox systems along the way. So none of these involve nucleases, things that cut DNA that are normally found in our cells. So for example, the Cas9 nuclease is not normally found in our cells, it's bacterial. The FOC1 enzyme is normally not found in mammalian cells as well, and the FOC1 enzyme is also used in zinc finger nucleases. So when we start looking about this, one of the things that we're not going to have time to talk about, unfortunately, is that when we go in and start talking about CRISPR-Cas9, in theory, as I mentioned, one of the techniques that you can use is to cut DNA and then replace that DNA that you've just cut with a working functional copy if you know that there's a mutation or you know that there's a deletion, etc. We're not going to have time to talk about this, which is one of the first points up here. The gene therapy tools we're not going to, unfortunately, have time to go over, but I will mention, for example, the different elements of CRISPR-Cas9, and in theory, you should be able to think a little bit more about this. So again, I'm going to go through some of the major therapeutic advantages, and I have to apologize ahead of time. I know that you don't have everything that's written down here on the right. I will make this slide available for you a little bit later on, and it is hopefully streaming for you as well. And these points are actually really important ones. How many of you remember from your first or second year biology classes the Cre-Lox system or the LOX-P system? Do you remember Cre-recombinase? Kind of. So Cre-recombinase, and the ones that are more important are the ones in A as well as the ones in C. In theory, using molecular biological tools, we can insert any sequence that we need to around a target gene of interest. And some of those sequences, which are very short, 34 base pairs in length, which is what this is, we can insert LOX-P sequences of 34 base pairs in length around a target gene that we know, that we want to be able to manipulate. In all of these instances, we need to know what a target gene is supposed to be. And so if we do so, you can see that there are two different orientations that this particular LOX-P sequence can take. There is this inverted sequence where they're mirror images, 5' to 3' and then 3' to 5' of each other. And this inverted sequence, when Cre-recombinase binds to both of these LOX-P sequences, it will actually flip the gene that's found in between them. Okay, and you might be thinking, why would that actually be of any use? You might actually just want to turn off a gene so that gene won't be available anymore. Another option in C is to completely eliminate or delete this particular gene. You might have a gene that, for example, for whatever reasons, has a mutation that is very, very deleterious. You might want to eliminate it. And so if we put those same LOX-P sequences, but not mirror images of each other, they're both 5' to 3' here and 5' to 3' here, both pointing in the same direction if you want to think of it in the vernacular, around the gene of interest. When LOX-P binds to this when Cre binds to this LOX-P site and this LOX-P site, what it will do here is the recombinase, the nuclease, the thing that cuts DNA, will cause a circular DNA to form around the gene of interest, the target gene, and then it will cause it to be completely eliminated from that sequence. So this is the way in which we used to, for a very, very long time, manipulate the genome. This was the crude form of a genome editing tool. This is what I was brought up with when I was doing my graduate studies, to look at Cre-Lox sequences, to insert in Cre-recombinase. And none of these, again, are found in the mammalian system. We don't have these. We don't have LOX-P sites. They don't exist. They're only found in bacteria. So even if you had them, you shouldn't worry about them. In addition to that, you don't also have the Cre-recombinase either. It's also something that you have to introduce. But if you have both, then you can actually introduce these types of changes to your genome. Another system that came along a little bit later, and is very similar to the zinc finger nucleases. So I'm going to combine the talins and the zinc finger nucleases together. When we started to look at other systems that involved genome editing, there were some other endogenous species that allowed this to happen. And one of the things that was realized is that there's this enzyme called FOC1. And this FOC1 enzyme is the nuclease. It actually cuts the target sequence that you want. So you would have to identify where you want to cut. Then you would have to design the proteins, and this is what makes this challenging. The bacterial proteins that guide this FOC1 nuclease to the right site is really difficult to design as well as engineer. And it's not a nuclease, it's not a nucleic acid. These are protein guides, and you have to build them sequentially as well as the FOC1. There's one other thing that makes this challenging. When we talk about CRISPR, it's just like one nuclease. Here, we have to actually have two FOC1 enzymes cutting on the opposite strands. And that makes it also really, really challenging. You have to design two of these things to be able to cut at the place in the DNA that you want to. Now, this is one of the reasons why CRISPR-Cas9 is so attractive. And this is sort of what I'm going to get into over the next few slides. And one of the reasons why CRISPR-Cas9 is so attractive are for precisely the opposite reasons that I'm talking to you now. Making proteins, making little peptide guides to design the FOC1 enzyme to target a specific area of your DNA to cut is extremely difficult. It is actually quite challenging to be able to bioengineer different peptides and to design these peptides for specificity. That's one aspect. So you can imagine if something is hard, it's also expensive. So talins and zinc finger nucleases are much more expensive and much more time consuming and much more overall complicated than designing a nucleic acid like the CRISPR-Cas9 system. The nuclease itself, we don't need two. We just need this one Cas9 nuclease. One system cuts on both strands of DNA, and so this Cas9 system, using a guide RNA that is much easier to design, makes this whole process much faster and much simpler, and by extension, much cheaper as well. So again, this is sort of the overview, and I'm going to go through this type of diagram several times over the next few minutes. You don't have to try to memorize everything on this slide right now. Hopefully by the end of a few of these different types of slides, you'll get the general feel for it. So this is what it sort of looks like. This whole unit is called the Cas9 nuclease. This whole unit itself cuts both strands of DNA. I don't need a FOC1 enzyme up here to cut and another FOC1 down here to cut. I just need this one Cas9 nuclease. And one of the things that really makes this work well is that instead of designing a peptide, I can design an RNA to help guide the Cas9 to the appropriate location. I don't need protein guides or peptide guides. I can just use a nucleic acid guide to be able to do this. So this makes it actually much more efficient in terms of targeting the particular copy of DNA that we want to be able to excise or to manipulate or to cause an insertion into. In case you were wondering what CRISPR actually stands for, it's this clustered, regularly interspaced short palindromic repeat. That's part of the guide RNA that we'll be talking about. And then the Cas9 is this CRISPR-associated nuclease 9 that was derived from one of the bacterial species, which is the nuclease that we'll be talking about. Okay, so this is the core component of the nuclease. So there are two different domains that are associated with the nuclease. There is an active domain RUBC as well as an HNH domain that you'll see typically in this configuration here. So one RUBC cuts one strand and the other HNH part of the nuclease cuts the other strand. You don't need two nucleases. They're built in together in this sort of configuration here. It's all you need in that one Cas9 enzyme, and it allows for you to be able to cut both strands using a single guide RNA. Okay, so again, not to go into a lot of details on this particular diagram. Hopefully, again, you'll get a better idea and sense of what I want you to take away from this lecture by the end. So again, we're simplifying the process. We identify a target sequence on the DNA that we want to be able to manipulate to make an insertion or to cut it or whatever we want to do. So this is the area that we want to create a break in. Maybe it has a particular part of the gene that is defective. Maybe we want to go in and replace it. Maybe this is normally important in creating a specific enzyme. We want the enzyme to stop working. We want to create a little break in it. Whatever the reasons are, we can create this DNA cleavage using this Cas9 system and a simple guide RNA to target that specific sequence. Again, making it really simple and easy to use. So the only thing that we really need to do is to be able to design a complementary guide RNA or an sgRNA. Sometimes you'll see it the other way as a gsRNA. And whatever it is, as long as we have that one single guide RNA that we designed against the DNA sequence that we want to be able to target and the Cas9 nuclease, then it should, in theory, cause a DNA strand break in the region that we specifically want to cause it to be broken into. This is much simpler than having to create these guide proteins that you see here. Each of these has to be built separately. You can imagine stringing together this large protein guide for this FOC1 enzyme and another one on the other strand for the other FOC1 enzyme to do exactly the same thing. And this is the reason why everyone wants to use CRISPR-Cas9, because it has a much simpler system. You don't have to design anything using proteins. You can design it using nucleic acids, which is much more easier. The other thing that is unique about CRISPR-Cas9, you can do one other thing that none of these other techniques can do. And again, this is a big advantage. So if you're keeping a tally, which you should, because you'll be asked, what are the advantages of the CRISPR-Cas9 system? You can actually do something called multiplexing. Can you guess what multiplexing is? Any ideas as to what multiplexing refers to, or have you heard it before in another lecture? Multiplexing. It's like going to the movie theaters and watching more than one movie, whatever the multiplex, cineplex, whatever it's called nowadays. Any ideas what multiplexing might be? How many genes can I disrupt at one time with this? One, right? So in theory, with CRISPR-Cas9, I can actually target multiple genes at the same time, or multiple locations on the same gene. I can't do that very easily with the TALENs, nor with the zinc finger nucleases. Multiplexing right now, at least, is unique to CRISPR-Cas9. I can target multiple sections on your genome and edit them all at the same time. It's just like letting them all go to work at the same time. So I won't go into all of the details, but one of the things that is limiting right now about CRISPR-Cas9, so it's not always the best, there are some disadvantages to this. People are trying to find ways around this. One of the things that makes this somewhat limiting, and it's not as big a limit as you would imagine, there's a specific sequence known as a PAM sequence that you have to have in the genome to be able to target a guide RNA to the area that you want to cut. So most genes have this, so it's not a big concern, but it is one of the limitations that we have right now. There also has to be a guanine that's found specifically at the 5' end of your guide RNA, which also makes it somewhat limiting in terms of what you can actually target. So that means that there is always sort of a restriction in terms of what is being targeted. In terms of TALENs, they're a little better right now, at least by most reports. If you target the same gene using two different systems, CRISPR-Cas9 versus TALENs, and just see which one is better, and then you do the entire genome sequencing, TALENs look like they're much more specific, at least for the time being. So there are less off-target effects. If I'm trying to target gene X, and it turns out that I've affected gene Y as well as gene Z, then that's an off-target effect. And right now, TALENs don't have these off-target effects as much as CRISPR-Cas9 does. Now, those are being worked on, and we're getting more efficient nucleases, etc. But part of that is because we have to engineer two different vectors for two different strands to the same location using FOC1 in the TALEN system, it's much more specific right now. The other advantages that TALEN has over CRISPR-Cas9 is that right now we can't do a single thing, or very little, limited amounts of things with mitochondrial DNA with CRISPR-Cas9. TALENs have been shown to be effective in targeting mutant mitochondrial DNA. So there are some advantages still to be using TALENs, and people still like to use TALENs even though they're more expensive, more time consuming, etc. Some of you might also be wondering, you might have heard in other classes that you can turn off genes by using a technique called RNAi, or RNA interference. Why not use RNA interference? Why bother doing all of this stuff that you see here. Why do genome editing, for example? Why not just try to solve everything with RNAi? It is very convenient. It works really well. It was a technique, for example, that we used to use a lot in the lab when I was doing my postdoc, because it was a really fast, easy, efficient way to do things. And it's been used a lot in vivo situations for gene manipulation. The one thing that you should take away from this, though, the effects of RNAi are generally speaking temporary. They're not long lasting, they're not permanent effects, and they're limited to knocking down only genes that are transcribed actively. And so this is one of the big limitations in RNAi. The CRISPR-Cas9 system, in theory, can actually, because how you cut and what you put back into that cut region can induce both gain as well as loss of function mutations. You can create a new mutation, or you can create a mutation that takes away the effect of that gene in a loss of function experiment. And so we know that CRISPR-Cas9 has the capacity to do both. And the RNA pathway actually tends to be the least high fidelity. It has the greatest amount of slippage. It's working on a very non-efficient, not efficient, a very non-selective way in many instances. And part of that is because due to its inherent nature, RNA itself has a tertiary structure associated with it. It likes forming these hairpin structures, and that often leads to off-target effects in addition to everything else that I've mentioned. So this is an important slide that I'll just point out to you. When we look at designing those guide RNAs, these guide RNAs have certain characteristic features that we need to have. So this is what's going to guide the Cas9, the nuclease, to the appropriate location on DNA that we want to be able to cut. And some of those basic considerations are that the total number of G's and C's that are found in the guide RNA, the base pairs, there has to be a certain percentage. Somewhere around 50% is usually pretty good. 40 to 80 is the broad range that you have to take into account. There's a certain number of base pairs in terms of the length, about 17 to 24 base pairs. And again, to eliminate those off-target effects, it's better to have more than it is to have less. So you need a minimum of 17 to 24, but if you have 24, it's always a better sort of guide RNA system. So in theory, right now, as long as you have this guide RNA, you can actually introduce this into an animal species. And in theory, as long as you have a Cas9 that you're also delivering with it, so for example, you have these guide RNAs that might be encapsulated in nanoparticles. You coat nanoparticles, nanobeads with the guide RNA that you just created at the top, and you inject this along with an adenovirus that might also contain other guide RNAs. In theory, you might be able to do a lot of different therapeutic manipulations on a mouse that already has the Cas9 nuclease encoded within it. Remember, Cas9 doesn't normally exist in mammalian systems, so this Cas9 mouse has in all of its cells a nuclease that's associated with it. You might look at immune system functions, you might look at the brain, you might look for cancer, etc. And the most interesting thing that was done recently in 2014 that gave a lot of scientists great hope for this is that injecting in this type of vector allowed scientists to actually target hepatic cancer cells that were growing, and it seemed to show that there was an effect, that guide RNAs and the Cas9 nuclease that were injected would actually be able to target different types of growing cancers as at least as a proof of function. And again, don't worry about trying to memorize this particular diagram and the different elements that are associated with it. It's just a proof of principle that in theory, as long as you can deliver a nuclease, as long as you have a specific guide RNA, that you can directly systemically in the blood do an injection and cause an effect on an organ system, which was shown back in 2014. So here's a couple of things to consider. I just told you that there are lots of technical considerations and we're getting better and better at this. Can we do this like really effectively? And this is the big issue. So right now, the biggest patent fight isn't really about CRISPR-Cas9 anymore. It's about who can make the more efficient Cas9 nuclease. And if you happen to go through the literature and you're really interested in the molecular biology of this literature, the big race right now is to try to modify this nuclease. So every publication that comes out that shows a modification of a smaller nuclease makes that a better system. Can you think of a reason why having a smaller nuclease is actually better for you? So the nuclease is going to be just as efficient as the old-fashioned Cas9, but let's say you come up with a genetically smaller nuclease. Yes?
Because Cas9 go through nuclear pores, smaller it is, and then easier it can go through nuclear pores to target the DNA.
Possibly. So your colleague at the front, her response was the smaller the nuclease is, the more likely it is to get through nuclear pores to be able to target the DNA. But you actually have a nuclear localization signal in there to target that into that region anyways, and that's not usually an issue. The RNA gets transported there. Any other ideas? Yes.
Smaller it is, so there's less off-target effects.
Less off-target effects. That's certainly something everyone's working on, trying to get the nuclease to bind more efficiently with the guide RNAs, and definitely one aspect. Any other ideas? So right now, and again, I don't expect you to know this, but this is sort of the limit that we have. You're injecting all of this into your mouse. You generally put this into a virus, which is what this diagram is showing. And the best viruses that we have now, the ones that we know aren't going to cause cancer in you, so you're not going to get injected by a virus, even if your doctor tells you, I'm going to inject you with a genome editing tool that's going to get rid of your cancer in your liver, but the virus might actually integrate into other parts of your genome that might cause you to get cancer in your brain, right? And so the best targeting tools that we have using viruses, in this particular case AAV or adeno-associated virus, we can only package a certain amount of DNA into those. So we know that viruses will infect cells, and as they infect cells, they'll carry the DNA, including CRISPR guide RNAs, as well as the nuclease into the cell that they're infecting. In order to package all of that, there's a certain limit that we can package into these different adeno-associated viruses. And that limit is actually being reached because of Cas9. The Cas9 is relatively big. The smaller the amount of genomic material that we can use to package into the adeno-associated virus, the better off we're going to be able to be in terms of doing multiplexing, where we have many of these guide RNAs. The other thing that makes this difficult right now is, in theory, right now, if you're doing things in the brain or elsewhere, you need to actually inject two guide RNAs, a guide RNA plus a separate nuclease because it's too big. And so you're getting two different viral injections, and that becomes a big issue, which is that point down here at the bottom. How do you know that both viruses are going to infect the same cell? How do you know that that cell over there isn't going to get your guide RNA and not get the Cas9 nuclease? And you need to be able to ensure that every cell gets both the guide RNA as well as the nuclease. And so we're getting really limited in terms of how we can package this all together to be able to infect cells to create effective genomic editing. And so this is one of those papers that Fang Zhang published in Nature Biotechnologies. And so one of the things that he showed was that you can do this not in theory anymore, but in a real intact animal, in vivo, and you can cause genome editing using a smaller version of this Cas9. The original Cas9 nuclease was actually derived from the strep pyogenes, and what he used was a Cas9 from a different species, in this case Staph aureus, which was much smaller, allowing him to package more into his adeno-associated virus. And the other reason, of course, to be able to do this is since he is in a patent fight, and since literally this will cause like billions of dollars to go either to MIT or to the University of California state system. In order for them to be able to win, they have to be able to demonstrate that they've actually made progress in this field in terms of their patents. And certainly every time another paper comes out from one of the groups versus another, it gives more support that their patent should be upheld. So we have a lot of different things that we went through today. And I know some of that was really technical. And I have to apologize that I went through a lot of slides without a lot of detail, and I'm going to try to make sure that all the detail that you need to know are on these slides here. So when you go through these different techniques, I want you to be aware of the different enzyme systems that are involved in these genome editing techniques. So FOC1, for example, zinc finger nucleases, as well as in talins. What the different specificity is derived from, whether it's guide RNAs or from protein or peptide modules, what the different limitations are, and certainly between talins and CRISPR-Cas9, you should know the limitations that exist between those two. And I talked to you in the last few slides about the limitations right now on the CRISPR system and how we design the guide RNAs. Make sure that you're aware of that. And then why are some of the reasons people are looking at smaller size nucleases? And if you can answer all 4 of those, then on this section at least, you'll do well, and you'll get perfect on this section. If you have any other questions related to this technology, please let me know. There are a number of labs that are actually doing this, and I'd love for you to get involved in some of that research as soon as possible at an undergrad level. And again, sorry, we're going to end just a little bit early, and if you're going off to tutorial, good luck in your presentations.